This is the overview of the organization, a little bit of how innate immunity contrasts to adaptive immunity, and how the microbiome relates to all of this. So there are numerous resources that will assist you in your efforts to learn in this space. These are Garland publications that we like, and we've used a lot of figures from them. The classic Janeway Immunology, which has grown from a small book to almost 1,000-page book, and then Param, which is certainly not Janeway light, but it really focuses on human immunity, and I think it should be on everyone's shelves. So I encourage you to use these. So the basic question that we start out with every year, and for those of you who have been here before, I would say that 2/3 of what we present today is the same as it has been for the past several years. And about 1/3 is morphing into some of the new and exciting work going on. And this is really the same. So the question is, you know, why do we have an immune system, and what is the teleology of this? You know, we live in a sea of bacteria. The simple answer that most people would respond to is it protects us against infection. And as you know, that's overly simplistic. We are outnumbered by the microbes mightily. And so we have to have a way of differentiating those commensals and those organisms that are actually symbionts with us from those that can cause great difficulty, infection, and death. I'm going to give a few book shout-outs here. So this just reminds me. So one of my favorite books of the year, which I've put in my Twitter feed and have posted in some blogs, is a book called I Am Multitudes. I Am Multitudes by Ed Yong. Anyone interested in the microbiome, this is a must-read. It's so enlightening of a book for even people working in the field of microbiomics. He's a very prominent science reporter, and it's largely non-human microbiomics, but everything from coral to bees to probiotics and beyond. So a great book shout-out. Anybody wants details, see me on the break. So the notion of the causality for having an immune system, you know, can be dated back a generation. Sir Frank McFarlane Burnett and Peter Medawar, who won a Nobel Prize, first posited this self from non-self. We develop our cells, we have forbidden clones, we have to get rid of them. It was quite an interesting and powerful statement moving forward of how the immune system can differentiate self from non-self. Obviously, at this juncture, quite simplistic because we're very complacent with a lot of non-self, and it was an incomplete answer. In the 1980s, the late Charles Janeway, in a very prominent editorial known by all immunologists called The Immunologist's Dirty Little Secret, questioned what was going on in the laboratory. It was well known at that time that to generate a robust immune response in an animal, not only did you have to expose it to an antigen, but you had to use what all of us recognize as Freud's adjuvant, this kind of gamish of oil and alum and other elements that seemed to stimulate the animal to make a specific immune response. It was just done. And there was very little self-questioning going on. He said, you know, there must be something that is turned on by this Freud's adjuvant. And as opposed to just writing an editorial, he actually went out and did the science. And this started the notion that there are receptor systems that see these danger signals from infections that kind of ignite the switch to generate an immune response. So it was more than just a foreign antigen. There was some other type of alarm system going on. This danger hypothesis reached full-fledged in the 90s, that Polly Matzinger, a great proponent of this, reduces that it's not just a danger from infections. There are danger signals internally that will light our immune system up. You know, when our cells die, they have to die an orderly death. And if they don't die an orderly death, that can ignite our immune response. We may precipitate urate crystals. And these crystals, not being infections, have, as everyone knows here, the propensity to ignite the innate immune system to develop gout, polyarticular gout, and if anyone's seen a patient with really bad polyarticular gout, they look like they have septic shock. And many a patient has languished in the intensive care unit with polyarticular gout until someone figures it out that there's not an infection and it's a kind of a SIRS-like syndrome. These receptors, which we now call DAMPs, and I'll go into in a little bit of detail, are the kind of the initial signals that something is wrong, something needs to ignite an immune response, and this is canonical in the organization of the integrated immune system. So as we look at the immune system, I still think it is quite reasonable to consider this in multiple layers. These are artificial layers, and they are not separate or distinct. If we are challenged, whether it be from a mosquito bite or from a cut or something like this, we have barriers that are designed to neutralize the threat, the danger, eliminate it with minimal collateral damage, and we want to do it as quickly as possible. So, you know, everyone knows that the experiment is a reality of life. If you work in the hospital and a patient has HIV infection and they're viremic and there's a cut and that blood is on your intact skin, the likelihood of you developing HIV and acquiring this infection is infinitesimally small, because we have integuments, we have antimicrobial peptides, we have outer barriers that are quite important. We obviously have in our respiratory passages, we have ciliary clearance that when they become defective, and cystic fibrosis, et cetera, obviously leads to tremendous complications. We also have pH barriers, et cetera, which are now being breached by obesity surgery and new spectrums of infections. Beneath this, but really not beneath this because anatomically it's involved, is this innate immune system that has both soluble and cellular elements, and we'll define these. These are designed to react very promptly. They are without memory signals, and they can be ferocious in their biology. And this gives the host a few days to do the same thing. Neutralize the danger, eliminate it, protect the host. No collateral damage, or minimal collateral damage. Yet, we are long-lived organisms, and as I will tell you that over a very long period of time, we have developed a final and more sophisticated, if you will, because it has much greater molecular specificity for danger signals, adaptive immune system. That really means lymphocytes and all their attendant biology that we'll talk about extensively today. And this is called upon if the first two layers of our defense fail. So this is the phylogenetic tree. And this is the sum total of, the holobiont of the Earth. And we occupy this little corner up here of vertebrates. And this little red dot is the only part of the living holobiont that has an adaptive immune system, which means that everything else, and here's the line for prokaryotes and then eukaryotes, is really innate immunity. So innate immunity is not puny. It's not ineffective. I mean, these organisms are think of a cockroach. They're doing quite good, and they don't have any of those T cells and B cells. They're going to outlive us for sure. So now let's look at an organization. So here's one way of thinking about it. Everybody now today, this is in the news, can recognize, see all these little stripes. This is the Asian tiger mosquito. This is Aedes aegypti, and this could be carrying a lot of bad stuff, right? Alpha viruses, dengue, flaviviruses, and if it penetrates the skin and takes a blood meal, there is intense activity of innate immunity, the outer barriers, antimicrobial peptides to neutralize this so that this infection, which is shown in the red line, which in this situation is taking hold, does not occur. In the first few days, it will invoke cells and soluble elements of innate immunity to neutralize this, and hopefully not need to elicit an adaptive immune response. But in this portrayal, and this could be chikungunya, the virus period of viremia is relatively short. A lot of effective of host defenses are thrown at it, but there is a robust antibody response and a cell-mediated immune response. So all elements of immunity have been activated. And then reactivation is the potential sequelae of this. So let's break this down and see how this works. So innate immunity, remember, a few decades ago, this was kind of the boring part of immunology. Nobody wants to be dealing with innate immunity. Today, this is the hip part of immunology. This is where everybody is going because there is so much excitement going on here and understanding that innate and adaptive immune systems are really artificial, and they are far more intertwined than we ever thought before. Canonically, no memory. You get that immunization, you make an antibody response, you come back, 6 months later, and you have this capacity to rapidly make antibody. That's adaptive immunity. Innate immune system is activated repeatedly based on these broad danger signals. It's early, and it contains both cellular and soluble elements. Thirdly, it is expressed in our germline. What does that mean? That means that, that the encoding molecules are directly translated via sequences in our own genome, as opposed to T cells and B cells, which undergo all this post-germline recombination and mutations to greatly expand the immunologic repertoire. The recognition capacity of the innate immune system is not fine, okay? This is not a fingerprint. You can take a protein, mutate a single amino acid, and one antibody can find the protein, and the other antibody will not see it, just by very small mutations. The innate immune system is more like facial recognition. It's kind of profiling. This is dangerous and this is not dangerous. Maybe I want to take that back to use the profiling thing here, but you get the idea for this. The outer integuments of the immune system, as I said, could be, you know, at one time before its sophistication arrived at understanding this level of immunity, we were thought of as kind of Teflon people, and it was really just being intact skin or intact barriers. Far more complicated than that, and there's an intense biology of antimicrobial molecules that are highly enriched in skin and other mucosal barriers that are involved in pathogenesis of diseases such as HIV, where these are very strong protections. You know, why is HIV more readily transmitted via anal intercourse than vaginal intercourse versus oral intercourse? All has to do with understanding the intricacies of these antimicrobial elaborations. They're also involved in local protection against cancer. The clinical caveat of this is that, you know, why do patients with psoriasis who have such bad breach of outer integuments very rarely get bacterial infections of psoriatic lesions? It is really uncommon. Whereas people with atopic dermatitis often will have secondary staph infections, et cetera. It's because of the differential display of antimicrobial peptides, the psoriatic lesion being highly immunologically rich and generating just so. These are some of the reasons why these defensins may operate, because they have this ability for this electrostatic attraction that can actually disrupt the membranes of invading pathogens and kill them. Here's just one molecule, beta-1 defensin, to see this. So this innate immune system, which is operating, you know, it's in the background. And I've given you some concepts of some cellular elements or other cellular elements, or soluble elements, I'm sorry, such as complement, CRP, other members of the acute phase reactants. There's also cells. And these cells are predominant cells in peripheral blood and expressed widely throughout tissues. And they range from early lines of defense, such as dendritic cells, to worker bees, such as neutrophils, resident cells, such as macrophages, and wandering monocytes, eosinophils, and basophils. As I will point out to you, and as you already know, these are not exclusively operating in an independent innate domain. They are intimately involved both through soluble communication as well as their cellular receptors with elements of adaptive immunity. How does that work? Well, dendritic cells, just as a definition, this is an entire area of immunobiology, but it's important to keep this in mind, because now there is there are some very interesting early therapies that have actually gone into human trials by modifying dendritic cells to generate tolerogenic signals for diseases such as rheumatoid arthritis, where this work is progressing, trying to develop tolerance to CCP, cyclic citrullinated proteins, to block the production of antibodies to such, or ACPA. These dendritic cells occupy first lines of defense. Some people like analogies, just talking about, I'm a wine taster. You should come to my blog, Wine and the Rheumatologist. It's on Rheumatology Room Now from Jack Cush. And the reason that we call them wine tasters because they occupy these peripheral tissues. They are challenged by a signal, determine whether it's dangerous or not. While they are not potent killers, they have the ability to break these signals down, process them, and then they travel to secondary lymphoid organs where they have the capacity to activate cells, and I'll show you that in the next talk. So while these cells do not kill, they are migrate, and they have the capacity to engage and activate adaptive immune systems. Two major classes, monocytoid and plasmacytoid. They have differential functions. In the skin, we call them Langerhans cells. So important player, you need to be able to recognize these and to at least understand their basic principles. Macrophages are resident in tissues. These are very potent cells at killing intracellular pathogens. In their nascent state, they are very potent at taking out the garbage of cell life and death cycles. In each tissue, they have a slightly different phenotypic and functional capacity. We call them everything from histiocytes to osteoclasts. These are all derivatives of monocytoid lineages. As opposed to the dendritic cells, which migrate, these are stationary. We need these. These are these are like the killing fields that are established within tissues. And they have the capacity to secrete cytokines and activate and reactivate cells of adaptive immunity. The field of macrophage biology has grown by leaps and bounds, and there are innumerable subsets, but we often talk about polarizing macrophages into those that are highly inflammatory, making all the cytokines that we think about in terms of TNF and IL-12 and the capacity to induce differentiation of T cells that we'll hear about a little bit later. But there are also immunoregulatory macrophages that if we polarize to this, serve to quell certain types of immune responses. The ability to make IL-10, TGF-beta, et cetera, are all emblematic of this capacity. Now, I thought about greatly expanding this comment about neutrophils, and we can either talk about this for a minute or take the rest of the day, because this is such an area of intense development. So we all understand that these are the most abundant cells in peripheral blood. We know that they only live a few hours unless they're activated, and they can hang around for a few days. They're very potent killers, and they have numerous effector functions. They're phagocytic. They can create these phagolysosomes to kill. They have both oxygen-dependent, oxygen-independent killing mechanisms. But these cells die at an astounding rate, hundreds of billions of cells per day. The body has to have great order. This has to be entirely orderly for this not to create its own danger signal for us. So everyone now understands the concept of apoptosis, programmed cell death. And this is the way that under physiologic circumstances most neutrophils die. Many of us recognize you have a pathology report that says this is leukocytoclastic vasculitis. What does that mean? That means that cells have not died in apoptotic cell death. That means that they have died by necroptosis. They have exploded. And now you can see this nuclear debris, this product of karyorhexis, distributed in the tissue. This is not good because that DNA with its attendant nucleoproteins create their own danger signals. And that's why vasculitis occurs. And so this is another extreme. Now, in the past 6 or 7 years, alternative forms of neutrophilic death have been described, and most people are probably familiar with the term neutrophilic nets. right? So who's familiar with neutrophilic nets? Okay, so Dr. Rigby, you can come up here and talk for the rest of the day on this. You can correct me if I'm wrong. So I'm trying desperately to get my head around this area. So when we say neutrophilic nets, that means that neutrophils have died, and as part of the death process, they have extruded decondensed DNA. This originally was described as a component of host defense against microbes. And indeed, certain microbes can induce neutrophils to undergo this type of cell death, extrude these decondensed chromatins, and as this chromatin comes out of the cell, it drags enzymes and antimicrobial properties, and it is a potent killer of these cells. So this is a defense mechanism against a bacterium. Well, in the past year, as it turns out, there are other circumstances where neutrophils can die and also extrude decondensed chromatin, but the pathway by which this occurs is totally different. So certain bacterium, and we're constantly battling the microbes in our environment and in our own body, and they're not our friends, they're not our buddies. If they have the opportunity to get the upper hand, they're going to take it. And so all organisms, whether it be viruses or phages or fungi or bacteria, they have their own defenses against our immune system. So some organisms have developed proteins called leukotoxins, which have the ability to cause pore formation in neutrophils, and they also extrude decondensed chromatin. It looks very much like the nets that come out of our own defenses when they are beating up these microbes. So this has been called leukotoxin associated hypercitrullation, and the details of that are not important. Still more types of neutrophilic death are being identified where mitochondrial DNA can be extruded and have the phenotypic appearance of neutrophilic nets. So what I'm telling you at the present time is that these forms of cell death of neutrophils are intimately involved not only in our host defenses, but in immunoregulation. And when these systems go awry, they have the capacity to ignite elements of adaptive immunity. And there's much going on in the pathogenesis of rheumatoid arthritis that we won't have time to talk about today. But if anybody wants to talk about it at flipped classroom time, we can talk about the latest pathogen in the rheumatoid arthritis pathogenesis called Aggregatibacter actinomycetem comitans. So it has 15 syllables, and we'll run out of time if we talk about it because it's so long. So let's move from there. Another area of great interest right now are these non-traditional lymphoid cells that go by many different classification schemes. So these are not T cells and B cells, and they kind of bridge the gap between innate and adaptive immunity. They are not T cells because they lack canonical T cell antigen receptors or have receptors that are not highly diversified, but these cells occupy first lines of defense, such as in mucosal surfaces and in skin, and they have the capacity to be polarized, as Dr. Looney is going to talk about, into cells that can secrete cytokines such as gamma interferon or IL-4 or IL-17. These have been incriminated in the pathogenesis of a variety of diseases, including the spondyloarthritis, psoriasis, inflammatory bowel disease, and we are very much closer to the beginning than the end of this story. So how do these cells recognize danger? By these molecular pattern readers on their surface. They can recognize that associated with pathogens in the bacterial cell wall. They can recognize nucleic acids that are not properly disposed. Remember necroptosis, karyophysis, and they can recognize garbage that is dangerous to us, such as uric acid. They all have the ability to recognize these repeated patterns. And here's a very nice display of, here's this molecule C1q. It always looks like a lunar lander. And the reason is that it can, this is actually mannose binding lectin. I said that, I described it as C1q because it has very structurally homologous appearance. And it can bind to bacteria that have these repeated glycoproteins on their surface, whereas humans do not. So these are naturally made to pick up microbes and protect the host. And that's just, you know, if it were only that simple, saying that number one. These cells of innate immunity are festooned with receptors that can bind to all these danger signals on both living organisms and dangerous garbage and ignite innate immunity. Many of them you recognize, and this, there are terms here that I'm encouraging. You know, this whole day is, as I say, it's not a definitive course. It is a framework for learning. If you find something that's of interest to you and you have param or you want to come back and read this, these are important. So the notion that there are families of receptors out there to recognize danger, the toll receptors that everyone's heard of, but also nod receptors that can be mutations of which lead to autoinflammatory diseases like inflammatory bowels, the RIGs, which were originally thought as antiviral receptors that have very broad antimicrobial capacity, and then a variety of other soluble elements that are all designed to be out there picking up these facial recognition patterns of danger signals that we are surrounded by. This large family of toll-like receptors, of which there are 10 variants, they have different jobs. Who recognizes lipopolysaccharide? Who recognizes flagellin? Who recognizes bacterial DNA or double-stranded RNA, which doesn't even occur in our host? So they all have respective jobs. Once they are bound, they then must transmit a signal to the nucleus to start a program of protein synthesis that will participate in the defense mechanism. I like to reduce this to two major cascades. One, which is to ignite an inflammatory pathway, and they all have the capacity to activate the nuclear binding program generated by NF-kappa B, which can generate hundreds and hundreds of inflammatory molecules. We've known about NF-kappa B now for over 20 years. So a toll receptor recognizing danger, igniting the fuse to send NF-kappa B to the nucleus and to make all of these inflammatory cytokines to initiate the immune response. Several of these toll receptors also have the capacity to trigger a second program, and those are interferon pathways. And as you well know, these interferon pathways are very prominent in antiviral defenses. So we're now starting to see some symmetry. Here we have the host. The host is, you know, happy sitting on that beach, that mosquitoes come along, penetrated the skin, taken a blood meal, penetrated the outer defense mechanisms of the skin, ignited via toll-like receptors, the innate immune response, because this mosquito is carrying a flavivirus or an alpha virus, and there is a local, regional immune activation. These cells that lie within the skin largely have innate roles. There'll be dendritic cells, there's resident monocytes, and the igniting of these inflammatory pathways now lead to the production of multiple cytokines that all have downstream biologic activities. Obviously, IL-1 can produce fever, TNF, which is involved in all types of homeostatic mechanisms at nanomolar or attomolar concentrations. When it becomes higher, seen in higher concentrations, can lead to systemic inflammatory reactions like SIRS, IL-6, the pivotal cytokine in both innate adaptive immunity as well as linking to viscerosomatic cells will cause hepatocytes to synthesize proteins in the acute phase program, and so you're going to see jumps in CRP and fibrinogen, and then other cytokines can lead to additional activities. So this is all fighting this virus at this level. Either we contain it at this time, kill it, or we have to call in more troops. This famous article, which has been read by so many people, has so many citations by Gabay and my dear friend Irving Kushner from 1999, really made us initially think about the biology of the acute phase reaction. As clinicians, we order these tests. You know, CRP's up, there's inflammation. Sed rate's up, it's inflammation. Fibrinogen. Each one of these molecules has a biologic role to play in this play that I'm outlining for you. You know, CRP has the ability to activate complement. It can bind to certain polysaccharides of bacterium. Haptoglobin is an antimicrobial peptide. Fibrinogen is involved in wound remodeling and reconstruction. Complement is an acute phase reactant. You want more complement because it needs to be activated either by classical or alternative pathways. Think of the acute phase reaction as an intense biological ballet. So this is where we are at this juncture. We have this danger signal that has been ignited the innate immune system. It's penetrated our integuments. This rapid response, fixed response, is now active. These effector mechanisms are in play, and I will tell you that this will be overwhelmed, and we will need adaptive immunity, and we'll talk about that in our next session. I'd like to take the last few minutes here to talk about something that is new and evolving. So that's really pretty much 101. You can go back and review it when the tapes come out, but this is really microbiomics, and this would be going up and up and up and up if I had more citations to show you that you can't, patients know what the microbiome, everybody knows what the microbiome is, and it is front and center in understanding basic and clinical immunology now. So we need to take a few minutes to kind of expand our horizons. The term microbiome, which was actually proffered by Lederberg, is really the sum total of the associated microorganisms, which it also is referred to as the microbiota, that we carry with us. So that is a term I want you to have. This is a glossary. I will use the term nonobiotics because we now understand microbiomics because we can examine animals that are raised in sterile environments. They have no microbes, at least prokaryotic microbes. And so this is a tool. Metagenomics is why we're here. We have the ability to understand the microbiome because of advances in sequencing and informatics that take in this amazing amount of data to make it translatable for us to study genes in our environment. A new term that I've added this year is holobiont. I've used it several times, and I've actually have become a student of this notion of the holobiont since reading I Am Multitudes by Ed Young. And I like it because it's really defined as the way I think we should be thinking about this. It isn't us and our microbiome. It's us with our microbiome. And they define this as the host plus all of the microbial symbionts, including those that are transient and stable. And this is not just bacteria, not prokaryotic bacteria and archaea. This also includes the mycobiome, all the meiofauna, the small primitive prokaryotes, eukaryotes, as well as the microvirome, and which we don't have time to talk about today, but we are one. So a few just kind of high-level altitude comments about the microbiome. This is a beautiful integrated review from the New England Journal by Lynch and Peterson from just a few months ago. If you really want to get back up to speed about this in 7 pages, this is extraordinarily good. And here what we see is the microbiome diversifying as a function of age. And first thing to point out, here's a person being born. A person is not born sterile. And in fact, as we now understand, the placenta has its own microbiome. This may sound weird to us, like it's a sterile environment. Well, we're just doing work at our institution on the vascular microbiome. Sterile aortas taken out and maintained in sterility have a rich microbial background. We are inundated with microbes that we're just starting to understand the relationship with. The microbiome diversifies over our pre-adolescence, and at each stage has different roles to play with different metabolic functions. Early on, it's thiamine, B vitamin metabolism. As we reach our kind of adulthood, we now have the ability to metabolize monosaccharides that have the influence to differentiate and influence immunologic development. And then we reach a plateau stage of all the time the microbiome is becoming more diversified. So there's a concept. Diversified microbiome is healthy. Losing diversity is often associated with disease states. And over time, as with everything else in our body, the diversity starts to contract, and this affects us metabolically. It is dynamic. Another concept, is there a normal microbiome? Everybody wants to say, I want normal, give me normal. And I'm going to go down and take those probiotics, and I'm going to just be there today. And there have been people that say that the only normal microbiome is, someone living, on a paleo diet in the bush. And there's an infamous microbiome scientist that did a fecal transplant with an Aboriginal fecal sample to himself. So this is living the dream. I don't know exactly what happened there, but that just showing you there's a lot of stuff going on here. So humans, we only have 20, 30,000 genes. But if we look at metagenomic studies of just the bowel microbiome, there are many-fold more genes than that. 3 million genes, and 500,000 of them are unique. They don't have homologs within the microbial world. This shared gene pool, as I will make the point in the last few minutes, is important for immunologic diversity and immunologic function and control. There is no such thing as a normal microbiome, but there are enterotypes which attempt to examine and analyze and present data as these diverse phyla, and they're pretty reproducible. The inter-individual differences, however, are dramatic. No two people have the same microbiome. This has been actually used forensically as well. If we just look at the gut microbiome, disruptions of the gut microbiome, when particularly losing diversity, can be manifested as many diseases. Now, inflammatory bowel disease we would get very easily, but there's dysbiosis in people that have atopic dermatitis. There is this notion that people have derided for some time of the notion of disruption of a microbiome leading to central nervous system perturbations. And it is real. We don't know how to define it. This notion that non-celiac gluten sensitivity in certain people can lead to profound disruption of normal higher cortical function is real. Everybody thinks they have it, but the outer boundaries remain to be seen. So just a notion, throwing that out. These are the tools for analyzing the microbiome. I don't have to go into time to go into them, but it's all advances in both sequencing and bioinformatics. That's the only reason we're talking about it. We're talking about looking at trillions of organisms and breaking this down into data chunks that actually can be analyzed. So this is from the Lynch and Peterson article. I referred to it. This is to remind me to set up the next few and final slides about the microbiome and immunity. If you take an animal, a nonobiotic animal, born sterilely, they're immunodeficient. They do not have a normal functioning immune system. Their Peyer's patches are diminutive. They don't have robust adaptive immune system. We need this microbial stimulation to develop a well-functioning integrated immune system. Why and what are the evidence? Well, first of all, there's preclinical evidence based upon numerous experiments, and I'm just giving you a few. So people try to manipulate the microbiome in animals to help you understand the influence on disease. And there's some very interesting work at Tarashi in Nature in 2017 showed that you could influence experimental colitis by giving a cocktail of organisms, and using 17 different strains of Clostridium. It's also shown that human microbiome and animal microbiome are profoundly different. You can't reconstitute a sterile mouse with human feces. They're still immune deficient. They need their own species type of microbiome to do this. And then multiple studies have demonstrated that bioengineered microbes in preclinical settings can actually be polarized to produce immunoregulatory cytokines to reset the immune system. So there's a lot of excitement about what might be coming down the pike. You can have designer microbes. How far are we in human microbiomics are there? Well, a few more preclinical notions. There are experimental models of arthritis that many people recognize, KBX and IL-1 RA knockout. they produce a very rich inflammatory arthritis, but these genetic models, if the animals are sterile, they don't develop inflammatory arthritis, only when they're colonized by bacteria. So there's a cooperation here that occurs. Now, moving from the preclinical to the clinical, we'll point out to you that in our next discussion here, my next talk on cancer immunotherapy and these remarkable advances with these checkpoint inhibitors that you see on TV and everybody here is well familiar with, as it turns out, one of the greatest biomarkers for how people respond to immune checkpoint inhibitors is their microbiome profile. And there are certain microbiomic profiles that correlate with a robust response to checkpoint inhibitors and other microbiomic profiles that don't. Finally, I'll point out this very important study in JAMA Peds from just a few months ago. It's called the TEDDY study, which showed that neonatal supplement of probiotics, just not fancy designer probiotics, but regular probiotics, if it was given in the first 30 days, showed a 60% reduction in the risk of pancreatic autoreactivity in high-risk children. So these people carried genes for type 1 diabetes when they were studied subsequently. If they gave the probiotic after day 27, they lost the effect. So in people, already been demonstrated, you can modify this. Whether they will have less diabetes or not, we don't know, but this is pretty important stuff. I'll point out one other study that I think is really worthwhile examining. And this is Fujimura from Nature Medicine from September of 2016, where neonatal gut microbiota associated with atopy and asthma. So what they did was they took almost 300 children and they looked at their microbiota at age 1 month. And then they divided them into 3 different enterotypes. This is how we classify bacterial pathogens. These are normal neonatal gut microbiomes. They could classify them into 3 different categories. And then they examined these children 2 years and 4 years later for atopy and asthma. And so looking at the gut microbiome at 1 month, had the ability to be associated with serum IgE to a specific panel of allergens. And here are the neonatal gut microbiomes. And this is actually looking at the phyla over here, just to show that you can see that there are remarkable differences. But just by categorizing these people, significant differences between IgE levels 2 years later by looking at their gut microbiome at 1 month. And this also correlated with some clinical parameters. Furthermore, when they took fecal water, put it through filters so it was sterile, and then incubated it with CD4 cells, these normal gut microbiomes, the atopic ones, actually cause more secretion of IL-4, which is associated with atopic disease, and also cause stepwise reduction in T regulatory cells. So the time is here. Just since the last time that we had our meeting here, we are now manipulating and analyzing the microbiome as a biomarker. I'm going to skip this and talk about, just make a couple comments about diet. So we all want to manipulate our microbiome. Is it possible? Well, there are a lot of data to suggest that we can. You know, our genes have only drifted less than 0.5% in the last 1,000,000 years, but our diets have changed 1,000-fold in the past 300 years. Our ancestors don't recognize what we eat as food for the most part. It comes in packages and sits on your counter for 6 weeks, and it's okay. There are receptors in the gut that are like toll receptors in the innate immune system. Here's just one bit of data. This from a very nice analysis from the New England Journal on aryl hydrocarbon receptors. These are found on cells of innate immunity in the gut. They can be triggered by phytochemicals that are found largely in the Brassicaceae family. It's like broccoli and Brussels sprouts and good stuff for you. And they can polarize these cells to make cytokines to actually lead to greater gut integrity, less leaky gut. There is a great step moving forward in understanding how food influences our immunity. And this is a slide that I probably showed before, but this is from our own Stan Hazen's work at the Cleveland Clinic, showing how the flora in the gut are involved in the metabolism of phosphatidylcholine that leads to the production of something called TMAO, which has been linked at both the preclinical, translational, and clinical level with atherosclerosis. So by modifying the gut flora, taking all the good stuff and cheeses and fatty foods, we are dependent upon this from happening. So I'm 4 minutes over on level 1. And here we are. We've had our drinking our Corona on the beach. We're bit by that Aedes aegypti, and now we're over here. We've overwhelmed our early defenses and innate immune system, and we need a boost.